The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our biochemistry focused webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in future webinar series. Please see the website for more details. My name is Dr. Robbie Baldock. I'm a senior lecturer at the University of Portsmouth and currently the chair of the Genes Research Area Committee for the Biochemical Society. Today's webinar is titled Developments in Genome Organization and is part of our dedicated early career research program of webinars. We are delighted to provide this opportunity today for Shifeng and Wilson to share their work with the molecular biosciences community. Before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. And if you have a question, please type it in the question box as shown on the image in your screen. Please also state who your question is for, and we will try to answer as many as possible at the end. Our first invited speaker today is Shifeng Xu from the National University of Singapore. Shifeng obtained her PhD in developmental biology from the University of California, San Francisco. She completed her postdoctoral training at A*STAR Singapore in human genetics. She is currently an assistant professor at the National University of Singapore, and she was awarded the Harold Weintraub Graduate Student Award in 2015, the Young Scientist Award by Singapore National Academy of Sciences in 2018, and the L'Oreal UNESCO for Women in Science Award in 2023. Her lab studies the epigenetic regulation in the epigenetic regulation in development and disease, with a particular focus on epigenetic regulators involved in rare genetic diseases. Today, Shifeng will be discussing her work entitled SMC-HD1 compacts DNA directly in an ATP-independent manner. So over to you, Shifeng.
Okay, great. Can you see my...
Yes, we can see your slides.
Okay, great. All right. Thank you. Thank you for the introduction, and thanks for giving me the opportunity to present our work here today. So today my talk will be on an epigenetic repressor called SMC-HD1. And let me change my pointer. And so today's talk will be on SMC-HD1 and how it can compact DNA. So as Robbie mentioned, my lab is interested in epigenetic repression. This audience should be quite familiar that there are many different epigenetic mechanisms that control gene expression. Some of these include DNA methylation, histone modifications, and the topic of today's webinar, which is genome organization. So today's talk will be on this protein called SMC-HD1 that we have been studying for a while now. SMC-HD1 is an epigenetic repressor. It was first discovered to be involved in the process of X inactivation. Mammalian females have 2 X chromosomes. One of it needs to be silenced, methylated, and compacted. But SMC-HD1 has been found to bind to the inactive X chromosome, keep it silent and compact. In knockouts of SMC-HD1, the female mice are embryonic lethal because they cannot do X inactivation. Beyond the inactive X chromosome, SMC-HD1 also have targets in autosomal genes. Interestingly, in humans, mutations in SMC-HD1 cause 2 different human genetic diseases. Heterozygous loss of function mutations cause a muscular dystrophy known as FSHD. So in this disease, there is not enough functional SMC-HD1. The loss of function mutations can be throughout the protein, here in this diagram. A few years ago, we found that very specific missense mutations in one domain of SMC-HD1 causes a completely different disease where patients are born without a nose. So it's really interesting to us how mutations in the same gene can cause very different outcomes. And that's why we're interested in understanding how this protein works. So what does SMCHD1 do? Most of the previous studies on SMCHD1 were in the context of actin activation, and work from many different labs have shown that when you lose SMCHD1 in female cells, there is a loss of DNA methylation on an inactive X chromosome. There is less spreading of marks such as H3K27 trimethylation, and there's a reduced long-range interactions. But how does SMCHD1 cause all these effects? And that's something that we wanted to understand. And the way that we went about doing this is first looking at the domains that SMCHD1 has. SMCHD1 works as a homodimer. So in this diagram over here, one monomer is on top and the other one is at the bottom. It has two recognizable domains. And I'll first talk about the SMC hinge domain on the C terminus of the protein. SMC hinge domains are found in SMC proteins, which form part of the cohesin and condensin complex. These are protein complexes that condense DNA during mitosis and meiosis. Cohesin has another important role in interface in the formation of topologically associated domains, or TADs. So the way these proteins work is that they bind to DNA and they actively pull DNA through the protein complex to form these long DNA loops. And this process is known as loop extrusion. This looping process forms these TADs interface. Because SMCHD1 has this SMC hinge domain, it's been classified as a non-canonical SMC protein. The other domain that SMCHD1 has is a GHKL ATPase domain on its N-terminus end. GHKL ATPase is a weak ATPase that's found in the MORT family of epigenetic repressors. These are proteins that silence transposable elements in animals and compact heterochromatin regions in plants. So the way that they work is that they're thought to many molecules of MORT proteins will bind to the DNA, and together they will compact the DNA into a tight cluster. Because SMCHD1 has these domains that are found in proteins that compact DNA, we wonder whether SMCHD1 can also compact. To address this question, we decided to take an in vitro approach with purified protein and DNA. And we collaborated with Professor Yan Jie from the Mechanobiology Institute here in Singapore. For most of our studies, we'll be using atomic force microscopy, or AFM. If you're not familiar with AFM, in AFM there is a very sensitive tip that we use to probe the surface of a sample. The displacement of the tip can be measured, can be detected by a laser. And doing this, we can get very high vertical resolution in the nanometer range. This is much higher resolution than any optical techniques. So using this technique, we can visualize individual molecules of proteins and DNA. Purified human SMCHD1 and visualized it by AFM. So if we zoom in on one molecule of protein, it looks a little bit like this. It's in a rod shape, very similar to how we draw our model. So on one end, there's the hinge domain, which is constitutively dimerized. On the other end are the ATPase. This is also very similar to a previously published negative stain electron microscopy micrograph of the mouse SMCHD1. So you can see that the mouse SMCHD1 also looks like a rod. So we know that our technique is working and we are getting the expected confirmation. So we do see many molecules of SMCHD1 in a rod conformation, which we call the closed conformation. But we also see molecules in other conformations. In some cases, the arms of the SMCH1 seem to be open. ATPase domains are very far apart from each other. We also see molecules in a P shape and in an O shape. We can image one molecule over and over again using high-speed liquid AFM. And doing this, we can see that the molecule is very dynamic in solution, and it can change between different conformations. Some still frames of another molecule, so we can see that it starts in an open conformation, and over a few frames, it becomes this P-shaped conformation. So what we're trying to show here is that SMCHD1 is very flexible. It adopts multiple conformations, and in solution, it is very dynamic. It can switch between the different conformations. So what does it do with DNA? We incubated SMCHD1 with linearized plasmid DNA that's about 3 kb long. And we saw the presence of these large clusters. If we zoom in into one of these clusters, we see many DNA ends that are coming out of the cluster. You can do a section profile through the cluster, and it's mapped over here. You can see that the center of the cluster is very dense, about 9 nanometers, which is much higher than the 1 nanometer of DNA, of naked DNA. So we think what's in the cluster is that many molecules of SMCHD1 has come together, and it has brought together a few molecules of DNA into this type. How is SMCHD1 able to do this? We try to capture intermediates of the compaction process. We see that in some cases, when the protein binds to DNA, it can induce a DNA bend. In other cases, we find that the protein is binding at the base of DNA loops. So we think what's happening is that SMCHD1 can bind to DNA and bridges the DNA to form these DNA loops in order to compact the DNA. For a protein to compact DNA, it needs multiple DNA binding sites, because if you're just holding on to DNA with one binding site, it's not possible to generate a force to compact the DNA. So it's been known that the SMC hinge domain is a DNA binding domain, but we don't know whether any other parts of this protein can also bind DNA. While looking for another DNA binding domain, we observed some EFM images where the ATPase domain of the protein seemed to be binding or in contact. And so we tested whether the ATPase domain can also bind DNA. So here is an EMSA assay with the hinge domain, which is our positive control. Hinge domain can bind DNA. And here we use the purified ATPase domain, and we can see that once again, this domain can also bind DNA. We confirmed this using AFM. Here we're looking at ATPase domain with DNA or hinge domain with DNA. And in both cases, we can see that the protein is binding DNA, as these dots that you see on the DNA. But these domains by themselves are not able to compact DNA into those type of clusters that we see for the full length. So here's a preliminary model. We think that SMCHD1 is very flexible in solution, so it can reach out and bind to DNA that's nearby. This helps to bridge DNA molecules together. As the local concentration of DNA increases, this attracts more SMCHD1 molecules to bind. This causes the formation of the tight cluster that we observed in our AFM. We think that the tight cluster may prevent the entry of other proteins, such as transcriptional activators, and that's one mechanism for how SMCHD1 can work as an epigenetic repressor. So this kind of DNA cluster has been previously seen also for cohesin. As I mentioned earlier on, cohesin is an SMC complex that usually compacts or moves DNA using ATP in a process called loop extrusion. But it's been shown that in the absence of ATP, cohesin can also form these large DNA protein clusters, very similar to what we have observed. And this process has been described as bridge-induced phase separation, that this tight cluster is now a separate phase from the surroundings. So as I said, this process by cohesin is ATP independent, and so is the cluster formation for SMC. So all of the EFM images that I showed you previously were all done in the absence of ATP. But we know that SMCHD1 is an ATPase. It can hydrolyze ATP. So what is the role of ATP in this process? And to address this question, we used a different single molecule assay. And that's the magnetic tweezers. And we switched to this because this allows us to have better temporal resolution over the DNA composite. So in this assay, we have DNA that is attached to a glass slide. And the other end of DNA is attached to a magnetic bead. So we can control the force on the DNA using a magnet that's above the DNA. Here's how the assay works. We first stretch out the DNA using a high force of 8 piconewtons. And then we lower the force to 0.5 piconewtons. This allows the DNA to relax. And after a certain period of time, we stretch it out again to 8 piconewtons. The DNA should go back to its original length. When we add SMCHD1 in, we see that when the force is lowered, the length of of the protein reduces, showing that there is DNA compaction. When we do the same experiment but in the presence of ATP, we see that there is compaction, but the compaction is less efficient. So each trace here represents one molecule. And we can quantify over here, over many, many different molecules, that in the presence of ATP, the compaction rate is lower. So here we can see two things. First, DNA compaction doesn't require ATP, similar to what we have seen in the AFM studies. And secondly, and more interestingly, ATP hydrolysis doesn't increase the compaction rate, it reduces the compaction. This is very different from any of the SMC compounds. So we wanted to follow up on this. We know that SMCHD1 is in contact with chromatin in the cell and not naked DNA. So we wanted to see whether these relationships also hold for chromatin. To do that, we made nucleosome arrays in vitro. Here's our nucleosome arrays. You can see that each individual nucleosomes are evenly spaced on the array. We incubated SMCHD1 with these nucleosome arrays to see if they can also be compacted by SMCHD1. So here are different AFM images going from left to right. We're changing the scan size. So the right is a smaller scan size, which you can think of as we're zooming in on the sample. And going downwards is different concentrations of SMCHD1. So you can see that first we do form these clusters just like what we saw with naked DNA. And secondly, this cluster formation is dependent on the concentration of SMCHD1. So with more protein, we see larger clusters. This is quantified over here. So if you can just remember this image over here at 18 nanomolar SMCHD1, I'm going to show you in the next slide what happens when we add ATP. So here, everything's the same. It's the same experiment. We're just adding ATP into the system. And what you can appreciate is that the clusters are much, much smaller. And this is quantified over here. So similar to what we see with DNA, when there's ATP hydrolysis, the cluster sizes are smaller. we perform a further bulk assay where we incubated nucleosomes and nucleosome arrays and SMCHD1 together. And we centrifuge it to separate the compacted nucleosomes from the non-compacted nucleosomes. We can then take the supernatant, which are the non-compacted nucleosomes, and run them on a gel. Here you can see that in the presence of ATP, there is more of the non-compacted nucleosomes. It's also quantified here in the graph. So this is showing once again that in the presence of ATP hydrolysis, nucleosome compaction is less. So here's our revised model. SMCHD1 is able to compact naked DNA as well as nucleosome arrays. Addition of ATP hydrolysis, we think is a regulatory mechanism to prevent the cluster from getting infinitely big. And so it probably reduces the cluster sizes to a physiologically relevant size. So here are our conclusions. We show that full-length SMCHD1 is very flexible in solution, and it can change its conformation in solution. SMCHD1 is able to compact DNA directly without the need of other proteins and without the need of ATP. Interestingly, ATP hydrolysis reduces the size of the SMCHD1 DNA clusters. One possible mechanism is by releasing SMCHD1 from DNA, preventing the clusters from getting too big. So it's been previously suggested that SMCHD1 may work as an insulator to prevent the binding of other proteins on its target gene. So one example is here. This is the protocadherin locus. In wild-type cells, there isn't so much CTCF binding. But when SMCHD1 is knocked out, then now we see a lot more CTCF binding. What SMCHD1 is doing is blocking this locus from CTCFX. So we think what we're seeing with the clusters, the tight clusters, may be a mechanism for how SMCHD1 is excluding the entry of other proteins. With that, I'd like to thank the people who did the work. All of the work was done by a very talented PhD student in my lab, Joelle, and with our collaborator, Yan Jie. These are my funding sources, and if you would like to contact me after the webinar, these are places that you can get hold of. And just one final plug, this is an early career webinar, and I just like to say that my department at the National University of Singapore is doing a faculty recruitment. We're hiring in all areas, but especially these ones over here. If you're interested or if you want more information, you can scan the QR code. Thank you, and I look forward to any of your questions.
Shifeng, thank you for a brilliant talk and for sharing some fascinating images there as well. Brilliant. So our second invited speaker today is Wilson Nartey from the University of Calgary. Wilson pursued his undergraduate studies in biochemistry at the Kwame Nkrumah University of Technology in Ghana, after which he received training in structural biology under the supervision of Professor Gerhard Gruber at Nanyang University in Singapore. Following the completion of his PhD, Wilson joined the laboratories of Arun Gudazi and Gareth Williams at the Ardie Charbonneau Cancer Institute, initially as a postdoctoral scholar and later as a research associate. Here he employs structural biology, biophysical tools to elucidate the mechanisms of actions of oncogenic chromatin remodelers in the context of cancer. Today, Wilson will discuss his work titled Mechanistic Insights into the Activity and Regulation of the Oncogenic Chromatin Remodeler, HELS. So over to you, Wilson. Thank you.
Thank you very much for the introduction, and also to the Biochemical Society for inviting me for this webinar. So today I'm going to share with you a story about a very elusive chromatin remodeler known as HELS. HELS basically stands for lymphoid. Excuse me. HELS basically stands for lymphoid. Sorry. my screen just here. HELS basically stand for Helicase Lymphoid Specific. Sometimes you also come across LSH, which means Lymphoid Specific Helicase. So when HELS was discovered about 2 decades ago, it was found to be overexpressed in the lymphoid tissue, hence the name. But over time, it became apparent that HELS is rather associated with tissues that are undergoing rapid proliferation. So there was also another name change to PASG, which means proliferation associated SNF2 like gene. Just to give you an example, I put this figure from the GTS portal, and as you can see, in most normal tissue, the HELS has been silenced, except in the highly proliferative ones like the testes, the lymphoid, etc. And to put it into context, let's compare that to CAD4, another chromatin remodeler. As you can see, not only the level of expression is quite different from that of HELS, the pattern also is different. And it is this characteristics of HELS to sustain proliferation that underline its oncogenic function. So far, we know that HELS play a major role in some of the most difficult to treat cancers. We are talking of glioblastoma, hepatocellular carcinoma, lung cancer, etc. In glioblastoma, for example, we know that HELS promote the stemness of the glioma stem cells by binding to the oncogenic transcription factors c-MEC and E2F3 to regulate the target genes. In hepatocellular carcinoma, HELS is able to reprogram the epigenetic landscape of most of the tumor suppressor gene, thereby promoting the growth and also metastasis of these cancers. So clearly, there is an urgent need to understand the mechanism by which HELS function. When I started working on this protein, very little was known about it. In fact, it hasn't been characterized yet, so I have to take time to characterize this protein. And through sequence analysis, what I was able to uncover is that at the very C-terminus, there exists 2 core regions that are quite distinct. So I named them core 1 and core 2. And as you see later on, they play different roles. And in the ATPase model, there is a unique insertion of about 60 or so amino acid right in the middle of the protein, which I will talk a lot about. And at the very C-terminus too is a motif, a unique motif characteristics of the HELS type of chromatin remodelers. So alongside the plant homologue, the DDM1, I went ahead, cloned and purified these proteins to perform some biochemical characterization. So using the ADP Glow assay, what we were able to uncover is that whereas the DDM1, the plant homologue, is stimulated, its ATPase activity is stimulated by DNA and the nucleosome substrate, HELS was completely insensitive to these substrates. And this also was reflected in the chromatin remodeling assay. So here basically we have a donor and acceptor flow for one at the end of the DNA and the other on the histone A2A dimer. So whenever you have chromatin remodeling, you lose FRET, and that's what we measure here. And as you can see, SNF2H has very robust chromatin remodeling, followed by the plant homologue, whereas HELS completely insensitive. So I went ahead to actually figure out exactly which of these unique motifs that I characterized earlier on is responsible for the autoinhibition. So with that, I performed a series of deletion, the N-terminus, the core region, the mid-loop, and the C-term. And as you can see, we were able to pinpoint the autoinhibition motif to this short piece here, the core 1. And we confirmed this using the kinetic ATP assay, NADH-based assay. We're also happy to find out that this activated ATP hydrolysis was translated to chromatin remodeling using this gel-based restriction enzyme accessibility assay. So in order to go a bit deeper, we really wanted to figure out how the COCO1 is auto-inhibiting health. So we knew that for chromatin remodeler to remodel chromatin, it needs to bind ATP and hydrolyze it. Secondly, it needs to be able to recognize the substrate, because I showed you earlier on, most of these remodelers require substrate to stimulate the activity. So to look at the ATP binding, we use a fluorescently labeled ATP analog known as TMP-ATP. And with that, we're able to show that both the wild-type cells and the COCO1 deleted cells both bind nucleotide to similar extent. So that was in the culprit. We next move on to substrate recognition. And here we compared the constitutively active DDM1 and HELS. In the absence of nucleotide, what we saw with DDM1 was that there is no binding to DNA. But the moment we add nucleotide, you can see there's a robust DNA binding over here. So basically what is happening is you have this N-terminus region of DDM1 sitting in the substrate binding cleft. And whenever you add nucleotide, it induces a conformational change that displaces this N-terminus. That was not the case for HELS, because what we saw was that in the absence of nucleotide, there was no binding similar to DDM1, but even when we add nucleotide, there was still no binding. The only scenario that we observed binding was to remove the COCO1, as you can see over here. And when we add nucleotide to this COCO1 removed HELS, the binding is highly improved. We saw similar situation with using nucleosome substrate, where the wild type failed to bind the nucleosome, but the COCO1 deleted HELS robustly bind nucleosome. So clearly, there has been some functional divergence when it comes to the N-terminus of these two highly homologous proteins. So we're quite excited by this discovery, because if you glance through literature, what became apparent is that almost all these HEL binders, from binds to HELs through the N-terminus COCO region. So we were sure that somehow to regulate HELs, one of these binders will be able to displace the COCO1. And indeed, that was what was observed around 2018, when the Funakabe lab showed that CDCA7, when partnered with HELs, formed this bipartite complex that activates HEL. So our work has provided that framework to understand how these binders activate or regulate the activity of HELs. More importantly, if you look through the cancer databases, sometimes we come across some structural mutation where the rearrangement so that the regulatory N-terminus have been replaced by fragments of other protein. And now we know, with our work, we know what the consequences of such mutation could be. So it's quite exciting time ahead. Next, I move on to the middle lobe. Remember the insertion in the middle of the ATPase model. As you can see from this alignment, this middle loop is only present in HELs, absent in all other chromatin remodelers. So we're quite interested to figure out exactly what it's doing. So this is an alignment of some chromatin remodelers bound to nucleosome that have been solved. And as you can see, this particular location where the middle loop is found is contain a very short variable region for most other chromatin remodelers, but for HELs, this has been replaced with a very large loop. So we decided to actually recapitulate this event. So what we did was we took the constitutively active DDM1 HELs, DDM1, and then we spliced this middle loop from HELs into the exact location where it's supposed to be, clone and purify this protein, and perform some biochemistry with it. So when we use DNA as a substrate, we were not able to observe significant difference between the ATPase activity between the wild type DDM1 and the DDM1 with health mill loop. However, when we use nucleosome as a substrate, we saw quite a significant difference between these two. And that reflected once again in the chromatin remodeling assay, where you have the DDM1 wild type here, but whenever you add, you use the chimera, you have almost a 4-fold difference. So we're quite excited by this, went ahead, solved the cryo-EM structure of health bound to the DDM1 mill loop bind to the nucleosome. And as you can see, the mill loop was binding to the A2A2 dimer, in what we normally term the acidic patch. And we're quite also excited that the recently released AlphaFold3 model of the full-length health bound to the nucleus was strikingly similar to ours. So let's zoom in a little bit into the this interaction. So in within this mill loop, there are 4 conserved arginines that we saw were interacting with the acidic patch residue. And to validate this, we generated 2 types of nucleosome, a wild type nucleosome and a acidic patch mutated nucleosome. And from our alpha screen assay, we're able to indeed confirm that by mutating this acidic residue of the nucleosome, we completely prevent the binding of the mill loop. And using competition assay, we validated the affinity, the EC50 to be about 40.4 micromolar. And the advantage of our chimera system is the ability to use FRET, and because of the sensitivity of the FRET assay, we're able to actually elucidate the contribution of each of the individual arginine, as you can see here. So there have been other structures of chromatin remodelers bound to the nucleosome, where some parts of these remodelers are engaging the nucleosome acidic patch. And as you can see, there's a lot of similarity among these structures. But what makes ours quite different is this induced turnover here. And this is because of the presence of these 2 proline, So we really was wondering, does conformational constraint modulate the binding of the mill loop to the A2A2B dimer? And indeed, when we mutated these prolines to alanine, we were able to reduce the affinity gain that we got from the chimera. Just recently, there was an in vivo studies that showed that within this mill loop, there is a serine residue that is highly phosphorylated. And this phosphorylation actually mediates the growth of lung cancer cells in vivo. As you can see here, this is the wild type. This is the wild type expression, tumor bearing the wild type cells compared to vector. You could clearly see significant difference. But when they mutated this serine to alanine, all the gain was lost. So somehow phosphorylating this mill loop is enhancing the activity of cells, which reflecting the expression profile of the target genes that helps regulate and overall growth of the cancer. We have the structure, so we wanted to actually look at the effect of the phosphorylation on the mill loop dynamics. So beside the serine 503 the group described, we also realized that there is a second serine 515, that is also highly phosphorylated. So using in-silico analysis, we're able to place this phosphorylate this mill loop and perform some molecular dynamic studies. So here in red is the dynamics of the wild type mill loop, but the moment you begin to phosphorylate it, you could see there is this conformational constraint that occur. And we have now accumulated data to show that this indeed leads to improved binding of the mill loop to the A2A2 acidic patch. So for the sake of time, I won't move to the C-terminus motif that I talk about earlier on. So as you can see, there's a lot of cancer-associated mutation in almost all the regions that I've described here. But this C-terminus, most of the mutations here has been associated to a syndromic disease known as immunodeficiency syndromeric instability facial anomaly syndrome That is a mouthful, actually. This mutation occurs within the C-terminal motif. The advantages of our chimera is that it gives us a very, very detailed cryo-EM map, allowing us to look at the interaction of the C-terminals motif, which is quite absent in some of the recently released DDM1 structures. So if you take a closer look at the C-terminals motif, you could see that this packing of the C-terminals onto the ATPase motor is being mediated, by this hotspot residue, this arginine, which is conserved across all the domains of life. And when we mutated this arginine to glutamine, what we saw was that it completely destabilized the protein, as you can see from the size exclusion here, and also this assay. So to understand what the role of this particular mutation associated to the ICF4 will mean for the stability of health, we look at our structure, and at this position, which is the leucine, which in the ICF4 syndrome has been deleted, we believe that by removing this leucine, we'll dislodge this highly important arginine from its pocket, thereby destabilizing the protein. So in conclusion, we now know that health exists as an auto-inhibited protein in what we term as the gated state. And this, in this state, you have the COCO1 blocking substrate recognition. And this gated state can loosely associate with the chromatin, but cannot form a productive complex. To form a productive complex, you need the presence of an activating partner, which is CDCA7, to come and engage or displace this COCO1, allowing the enzyme to engage the nucleosome. Once engaged, it then uses its middle loop to bind the acidic patch, stabilizing the histone A2A2B within the nucleosome to allow efficient translocation, DNA translocation to occur. So with the COCO1 ungated, middle loop engaged, you can now have a robust chromatin remodeling that will allow biotic recombination, DNA repair, mitigation, and also transcription to occur. So with this, I will say thank you for listening, and also thank you to my supervisors, Dr. Garrett Williams and Dr. Dazi, also Daniel in the Sharmes lab, and also Claire Atkinson at the UBC Cryo-EM Center for assisting in the data collection for the Cryo-EM data, and also the funding sources. Thank you very much.
Wilson, thank you very much for that detailed overlook of HEL's activity there. Fantastic. So we will now welcome questions from the audience for any of the speakers. If you have a question, please type it in the question box as shown in the image on the screen. So I think we've had a couple of questions already. If I could start off with a question for you, Shifeng. We've got, oops, oh no, I've lost it now. What is the effect of loss of ATPase activity in SMC-HD1 in mammalian cells? What is the phenotype effect of such a mutation?
Yeah, so all of our work with that was done in vitro with purified DNA, purified protein, or nucleosome arrays. But we know that in the cell, if there is inactivating mutation in the ATPase domain, SMC-HD1 doesn't localize onto chromatin. So I think in addition to whatever it's doing once it gets onto chromatin, the ATPase is also having a role in somehow localizing or recruiting SMC-HD1 to where it's supposed to bind on the genome. That's something that we haven't touched on in any of our in vitro studies.
Thank you very much. So second question goes to Wilson, if that's all right. Is there a link between HEL's activity and lung infections with cancer-causing viruses?
Yeah.
Perhaps speculative.
Yeah, that's extremely speculative. I don't think I can answer that with the data that we have now. But what we know is that overexpression of HELS in lung cancer modulate its growth and spread throughout the body.
Thank you very much. So I'm just going through here. Okay, so another question for Shifeng, if that's all right. They say, thank you for a very insightful talk. Could you elaborate on the feasibility of targeting SMCHD1 for treating FSHD? I'm going to abbreviate that for the sake of time. And how effective is CRISPR-Cas9 in vivo editing in potentially correcting this gene to address the disease?
Yeah, so there are quite a number of developments, therapeutic developments for FSHD. So as far as for doing genome editing on this protein itself, I think that has not been in any of the lead therapeutic developments, because for genome editing to work for muscles, you will need to target a lot of different parts of the body. It's very hard to deliver something effective to all your muscles with genome editing. But there are other therapeutic developments in the pipeline, such as doing things like CRISPR-I. So the reason why these people have FSHD is because SMCHD1 is now not active, so there's a toxic gene that's being overexpressed. So there's a lot of activity in trying to downregulate the toxic gene, for example, using antisense oligos or using CRISPR to genetically reduce the expression.
Thank you. So actually another quick question for Wilson's come through. So it was really interesting that you showed that the coiled coil domain has this auto-inhibitory function. So do you see mutations in those regions leading to a constitutive activation of HELS? And then the follow-up question is, what are the downstream impacts of that change?
Yes. So yeah, that was a very good question. Yeah, we've actually, as a follow-up to this project, we've actually performed some mutations within this domain, and we've seen it activating. And most of these mutations have been found in the cancer database. If I can share my screen quickly. Do you see my screen?
Don't know if we've got the ability to do that at the moment.
Okay, it's okay.
But if you're happy to give us an explanation. Oh, there we go.
So as you can see, all these clustered mutations are cancer associated. Okay, and we've been able to show that some of them, especially this T27I, leads to activation of the protein. And to answer the follow-up, so what happened is that whenever you introduce this mutation, it liberates, it sort of generates this steric hindrance with the motor, thereby liberating the COCO1 from its auto-inhibited state, leading to productive engagement with the nucleosome.
Thank you very much. So next question, we have one for Shifeng again. Thank you for the lovely talk. Do you have an explanation as to why ATP hydrolysis reduces nucleosome array compaction? You've reported that this mechanism is important for regulating cluster size, but why is it particularly relevant in the presence of ATP?
Yeah, it's a very good question that I don't have a good answer for. So our speculation is that ATP hydrolysis releases the protein from DNA. So as it continuously hydrolyzes ATP, then it will come off the DNA, and ATP leaves the protein, and it can bind the DNA again and do its bridging thing. So that's why in the presence of ATP, then the clusters that it's forming are smaller. As to why it has to do that. We don't really know. We just think that maybe it's a way for it to prevent clusters from getting too big, and unregulatedly big. So we don't have a good answer for that right now.
Okay, brilliant. So another question for Wilson. What is the next phase of your research, especially as it relates to the metastasis of cancerous cells?
Yep, I don't know whether I can share a lot about that, but yeah, we are actually following up with this work where we've generated a couple of lentiviruses, and we are performing some experiment just to look at the role of overexpression, both the wild type and also the activated, in the growth of these cancers and also their metastasis. We will do a whole slew of assay to ascertain that.
Thank you. We have another question for Shifeng, if that's okay. What would be the repercussions of SMC-HD1 functioning in an ATP deficient environment? Could the resultant larger DNA clusters contribute to any particular genetic diseases or disorders that you know of, or impact on genomic stability in individuals with those lower ATP levels?
Yeah, that's a very good question. So when there's less ATP, then we'll expect the clusters to be bigger. So maybe it's now repressing more things than it's supposed to. I don't know of any diseases that involve SMC-HD1 that has to do with more repression. So that's something that we can look and see if there's any link with that.
Thank you. And there's another question for Wilson that's just come in. You showed that there was phosphorylation in the mid group. What was your prediction on the impact that would have on the interactions or that would regulate function?
Perfect. We predicted that it will enhance affinity. In fact, that's what it did. You know, we've accumulated a lot of data that has shown that indeed there's enhanced affinity. We are validating it. That's why I did not show it. So basically, this technique is a strategy that has been used by medicinal chemists for years in what is known as staple peptides. So whenever you have a macromolecule that undergoes a lot of flexibility, and upon binding, there's sudden reduction of this flexibility. There's an entropic cost to it. So what happened with this phosphorylation is that it constrained the peptide movement so that upon binding, the entropic cost will be lower. And that overall leads to enhanced affinity. So this is a method, it's a strategy that the cell is using to actually tune the activity of health. Okay, so if you want to enhance the activity of health, put phosphorylation there. If you want to, tone it down, remove the phosphorylation. So we're actually looking for these kinases, and this phosphatase is responsible for that. And we believe that those could also be targets for therapeutic intervention.
Thank you. And there's another question for Shifeng, and we'll probably make this the final question, I think. You mentioned that SMC-HD1 silences transposable elements. Is this silencing a general effect or specific to particular subclasses, i.e. retrotransposons, DNA transposons, and what would the mechanism be? Let's make that a key question.
Yeah, SMC-HD1 doesn't silence transposable elements. It doesn't have a specific function in silencing transposable elements. It silences stuff on chromosomes, but those are not specifically the ones. Not any particular classes and not all of them in general.
Perfect. All right, thank you very much. I think that's all we have time for in terms of questions. That leaves me to thank everyone for attending, especially the speakers. So thank you very much for your time. You can continue the conversation online by following @BiochemSoc and at PP Publishing on Twitter. We, of course, would welcome suggestions for future topics and speakers to feature in the Biochemistry Focus webinar series. If you do have an idea for a webinar in 2024, we'd happily invite you to submit a proposal for an upcoming webinar, and you can find more information about the upcoming webinars, propose your own, and also watch previous webinars at www.biochemistry.org. All of the upcoming webinars are listed on the website. If you've missed any of the 80+ webinars that we've already run as part of this series, or would like to watch them again, please visit the website or the YouTube channels. So the details for that should be shown on screen. The recording from today's webinar will also be made available to you to watch within the next couple of weeks. Important points to mention: the Biochemical Society 2026 awards are now open for nominations. This features a wide range of recognition opportunities and a streamlined nomination process. So there's never been a better time to nominate an outstanding peer or colleague. The deadline for initial nominations is the 1st of November, with submissions welcomed by and for both members and non-members. So please visit the website to find out more. Finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. You can join the Biochemical Society's community of research and specialists and take advantage of key benefits, which include discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, and personal online access to 2 of the Portland Press journals, as well as many more benefits. Visit the website to find out more, and thank you all and goodbye.